very good evening to one and all present here. I am Srijan Sen, and today we'll be taking the week 9 live class session on the course Introduction to Complex Biological Systems. Okay, so welcome to the class. So I'm sharing my slides with you all. So basically, in today's class, we will be talking about infectious diseases. That's on virus and bacteria. Okay, right. So basically, when we look into the microbial world, the first thing that comes to our mind is basically the word microbes, which is defined as organisms and acellular biological entities that are too small to be clearly seen by an unaided eye. So you can categorize the studies. So microbiology study both the cellular and the acellular entities, and over there you see evolutionary relationship where you see the diagram of the universal phylogenetic tree, noting that these evolutionary relationships are based on RNA sequence comparisons. So looking into the overview of history of life on Earth, what we see over here is this is the microfossils of an Archean apex chart of Australia. So the first appearance of multicellular eukaryotic organisms came 1.2 billion years ago. Then the eukaryotic cells started to develop, and these were distinguished by a defined nucleus and a membrane-bound organelles. Then comes the fossils of the primitive filamentous microbes, which represents one of the earliest direct records of life, and the first appearance of prokaryotic cells. And over there you see the microfossils of an Archean apex chart of Australia. So these fossils were approximately 3.5 billion years old, and they have a preserved inchet. That's a hard, fine-grained sedimentary rock. And so these ancient microfossils are remarkably similar in structure to modern filamentous cyanobacteria, which is famous for the oxygenated Earth's atmosphere through photosynthesis. The early cellular life, where you had the archaea and the bacteria, which shapes our environment and drives evolution of complex life forms. So early Earth was a hot environment that lacked oxygen. So previous class we discussed on this. So there are heat-loving archaea capable of using inorganic molecules such as the iron sulfide as the source of energy, which is more likely a remnant of the first form of energy metabolism. So another metabolic strategy that is right. So another metabolic strategy is oxygen releasing photosynthesis appears to have evolved as early as 2.5 billion years ago. So fossils of cyanobacteria like cells found in rocks and dating to the time supports this hypothesis. So the oxygen that released is thought to have altered Earth's atmosphere to its current oxygen rich state. Now, what we understand also is that mitochondria and chloroplast contain DNA and ribosomes. Both are similar to bacterial DNA and ribosomes. So, both organelles belong to the bacterial lineage based on the rRNA analysis. So, replication of these two organelles is like bacterial replication, the binary fission. And the genome sequence of Rickettsia prowlesii, an obligate intracellular bacterium that causes epidemic typhus like fever, headache, rash, is most similar to the modern mitochondrial genomes than any other bacterium. So the microbial life under Earth. So Antoine van Leeuwenhoek is best known for his work in microscopy and for his contribution towards the establishment of microbiology as a scientific discipline. And he first observed bacteria through a microscope. So the image that you're seeing is the mold sporangia that was drawn by Robert Hooke in 1665 from his observation of objects under the microscope. Now, the foundation of modern microbiology comes where Robert Koch devised techniques of pure culture to study a single species of microbe in isolation. So his postulates also provide a set of criteria to establish a causative link between an infectious agent and a disease. Then you have some other scientists, Edward Jenner, who established the practice of vaccination. Then Louis Pasteur, where he developed the first vaccines based on the attenuated strains like the rabies vaccine. Then comes Alexander Fleming, who discovered the penicillium. Mold generates a substance that kills the bacteria. You have Florey and Chain, who purified the substance as penicillin to save thousands of lives. Now, microbes we know cause diseases, and some of these have a huge impact in the human history. So there was a plague in 1345 known as the Black Death that struck Europe with brutal force and like killing one third of its population within 4 years. Now the Black Death was caused by the bacterium Yersinia pestis that's typically transmitted to humans through the bite of an infected flea. Now, in 1918, the flu pandemic, the deadly influenza pandemic that killed millions of people worldwide. You have the cholera, the severe diarrheal illness that's caused by the Vibrio cholerae. So, cholera was a waterborne disease. And you have the smallpox, which is highly contagious and deadly viral disease caused by variola virus, eradicated by vaccination. Now, the key benefits of microbes includes that microbes are utilized in the production of fermented food like the yogurt, cheese, and bread for a very long time. The digestive health, we have the gut bacteria that breaks down the complex food components into nutrients that can be absorbed by the body, playing a vital role in digestion, right? Now, vitamin production, there are certain microbes in the gut that synthesize essential vitamins like vitamin K and B vitamins. The immune system support by interacting with the immune system beneficial. The microbes help train it to recognize and fight off harmful pathogens. From the point of view of an environmental decomposition, microbes play a crucial role in breaking down organic matter in soil, which is essential for nutrient cycling and maintaining the ecosystem balance. And bioremediation that contains microbes like the Pseudomonas and the Bacillus, which can be used to clean up pollutants in the environment like the pesticides, oil spills, etc. So from here we need to get into the concept of bacteria. So what does this say? So bacteria, these are significantly larger than viruses. Like you have E. coli that is rod-like shaped, measuring like around 4 microns. You have the streptococcus, and there are certain large viruses like the pox virus, influenza virus that is more of roughly spherical, measuring 85 nanometer in diameter. Then there are certain bacteriophages and small viruses like the T2 E. coli bacteriophage, the tobacco mosaic virus, the poliomyelitis virus. So poliomyelitis virus is one of the smallest known viruses, measuring only 27 nanometer in diameter. Now you can see over here there are certain other bacteria that are given. So you have the cocci, so also it's known as the coccus. So these are spherical or oval shaped bacteria, namely their name comes from the Greek word coccus, meaning berry. Depending on how they divide and stick together, they form different arrangements like the diplococci, streptococci, staphylococci, tetrads, sarcinae. So there are different types that are available. On the other hand, bacilli is basically rod-shaped bacteria. They resemble little sticks or cylinders. So common arrangements among them includes the single bacillus, the diplobacilli, the streptobacilli, coccobacilli, and the spiral and other shapes mainly includes the vibrio. There's a curved rods that looks like commas. Then you have the spirillum, thick, rigid spiral shapes, and the spirochete. So thin, flexible, corkscrew-like spirals. So the bacterial cell organization is somewhat like this, where you have, so starting from the leftmost side, you have the capsule that's present over here. These are some of the ribosomes. Over here you can see the cell wall, the plasma membrane that's present over here. As you move towards this side, this is the nucleoid. You have the flagellum that's sticking outside. This is basically the inclusion body. Then the spiral material is basically the chromosome, and the hanging tail or this wiggling like things are basically the fimbriae. Okay, so bacterial cell membrane and uptake of nutrients happens through the gated channels, and that's present through this cell membrane and the cell wall. So you see over here is a fluid mosaic model, and there are certain phospholipids, haploids. Okay, so there is a mechanism of nutrient uptake that happens. So you have heard of the passive diffusion, then facilitated diffusion, active transport, group translocation. So there are different ways of how they take up the different nutrients from the outside through a concentration gradient. And the key functions is basically to understand the selective permeability of the cell wall. So you should know what controls what enters and exits the cell, as well as there is an energy production like an ATP production is required very in the electron transport chain, since they lack mitochondria, and sensing the environment is also important because the receptor proteins on the surface allows the bacteria to detect and respond to chemicals, and that's known as chemotaxis. So yes, so there are different ways of how the nutrients are taken up. Now there are some unique features of the bacterial cell wall. So the gram-positive cell is basically known for its thick peptidoglycan layer, which is the most prominent feature. It is thick, multi-layered, has a meshwork of sugars and amino acids that provide the structural strength. Then you have the teichoic acids. These are unique to gram-positive bacteria. It does not have an outer membrane. So unlike the gram-negative bacteria, there is no lipid membrane outside the peptidoglycan layer. So staining, because it's critical to understand about the staining of a gram-positive versus a negative bacteria. So because of the thick peptidoglycan, these cells retain the purple crystal violet stain during a gram stain test. But on the other hand, when you look into a gram-negative bacteria, so the peptidoglycan layer is much thinner. So the peptidoglycan layer sits in the periplasmic space, that's the gap between the inner and the outer membrane. You have some of the liposaccharides, like these are found on the left outer leaflet of the outer membrane. They are important for the bacteria's defense and can act as the toxins in humans. Followed by, if you look into the staining, so it cannot hold on to the stain. So you have to stain them with safranin if you want them to be well stained. So these are different stains that are usually happening. So there are different steps of how you stain it. So crystal violet, you stain them for crystal violet for 1 minute and then rinse with water. Iodine is used as a mordant for 1 minute and then you rinse it. So alcohol as a decolorizer for 10 to 30 seconds and you rinse it. So what happens? So, and then you counter stain it with safranine. So what happens? The gram negative, because the crystal violet stain is not retained, it can take up the safranine stain over there. You know it's a gram negative bacteria. On the other hand, when it takes up the stain, it's a gram positive bacteria. Right, this is the bacterial cell wall and its unique features. You already I've already spoken about it. There is not much things that we need to discuss over here. Maybe a few things like the NAG and the NAM, that's the glycan backbone. So it's a polymer of alternating sugars. NAG, that's N-acetyl glucosamine, and NAM, that's N-acetyl muramic acid. Right, so the key features is that the NAG and the NAG, these specific sugars are unique to the bacterial kingdom. So D amino acids, so the presence of D-alanine, D-glutamate is a signature of bacterial only trait, and the DAP, that is the diaminophilic acid. So it's an amino acid that's not found in proteins, but it's essential for the direct cross-linking in many gram-negative bacteria. Okay. Now, the bacterial cell organization has certain special features like the bacterial cytoskeleton. So, you have the FTSZ that helps in cell division and is widely observed in bacteria and the archaea. The MRIB or the MBL, that's maintaining the cell's shape and localizes proteins and is present in most rod-shaped bacteria. The magnetosome, so small structures primarily made up of the magnetite and helps in navigation through the Earth's atmosphere magnetic field. And the MAMK cytoskeleton protein that resembles for the orientation of these magnetic particles. Now, when you look into the other special features where there are capsules and the slime layers, the flagella that helps in the movement of these bacteria. So there are certain motor proteins that creates a channel through which the protons can flow, and this causes the flagellum to rotate. There is an external energy that is also required for it, and then there is the endospore formation. Now, if we get into the horizontal gene transfer mechanism in bacteria and its implications, so there are three terms which we need to understand. One is transformation, conjugation, and transduction. So what does these three terms basically mean? So transformation is a process where a bacterium takes up a DNA fragment, thus an outer DNA fragment directly from its surrounding environment. So for this, you require the recipient cell must be a state of competence for the able DNA to pass through its cell wall. And the source that the DNA comes from, other bacteria that might have died or lysed at its burst. The conjugation is often called mating of the bacteria. So this involves a direct transfer of DNA from one live bacterium to another. It requires physical contact, usually through a protein tube called a pilus. And this is the most common way bacteria spread the antibiotic resistance genes quickly through a population. Transduction is a genetic material moving from one bacterium to the another through a virus, specifically a bacteriophage. So when a virus infects a bacterium, it can accidentally package some of its host DNA into the new viral particles. When those viruses infect the next bacterium, they inject the previous host's genes instead of viral DNA. And why does this matter? So the implication is that there is an antibiotic resistance. It allows the bacteria to survive in the harsh environments like adaptability, and is also useful for genetic engineering. So viruses are acellular infectious agents. They are non-living phase and living phase. So viruses could be considered as a simplest form of life, hence ancient, composed of nucleic acids and proteins. So its virus is more of an infection, obligate and intracellular parasite. It can exploit all three domains, archaea, bacteria, eukaryotes as their hosts, and they are submicroscopic particles. The general structural properties of viruses is that a complete virus particle is called a virion. So virus is comprising of genetic material which is surrounded by a protein coat. In some cases, an envelope is also present, so derived from its host cell membrane. And there are some enveloped viruses like the influenza viruses and the coronaviruses. And the non-enveloped viruses like the adenovirus and the poliovirus. So unique features of viruses is that virus possesses DNA or RNA as its genetic material. It lacks ribosomes or any membrane-bound organelles. And virus hijacks host cell machineries for their own good to make more and more virus particles. Now, viral multiplication happens through quite a few stages. So if you look at them quite step by step, so the first one is the attachment of the virion to the host cell. So where the cycle begins when the virion makes contact with the specific target cell. This is a highly specific process where viral surface proteins binds to complementary receptor sites on the host cell's membrane, much like a key fitting like a lock. Then there is the entry of viral nucleocapsid. So once attached, the virus enters the cell, and this can happen through the various methods. First one is endocytosis. It can be through fusion. So endocytosis, the cell membrane surrounds the virus and pulls it into the vacuole. And the fusion is for the enveloped viruses. The viral envelope fuses directly with the host membrane, releasing the nucleocapsid into the cytoplasm. And then the synthesis of the viral proteins and the nucleic acids takes place through replication and translation. Then there is self-assembly of the virions. So the newly synthesized genetic material and the protein subunits comes together to form new complete virus particles. This often occurs in a host cell cytoplasm or the nucleus. And finally, release of the progeny virion. So it's multiplication. So now it's time for the new viruses, new progeny to infect the neighboring cells, which happens through lysis and budding. Fine. So these are the multiplication of the viruses, the different steps which we just discussed. Now the types of viral infections and their effects on animal cells. So we understand how they enter and are actually multiplying. So like if we look into over here, so there are four ways of how it can actually affect into the animal cells. The first one is the lytic infection. That's the cell death. So the virus enters the cell, undergoes rapid multiplication, and eventually causes the cell to burst. That's lysis. And this results in cell death and viral release, allowing the new virions to infect the neighboring cells. You get into the latent infection. That's a delayed activity. So after entry, the virus undergoes activation, and then the viral genome remains part or present, but the host is not harmed for a particular period. So the virus exists in a dormant state and does not produce new viruses or kill the cell until it's triggered to enter the lytic cycle later. If we get into the third one, that's the persistent or the chronic infection. That's a slow release. So instead of killing the cell directly or quickly, there is a slow release of virus without the cell death. And the host cell remains alive and functions as a virus factory, budding of new viral particles over a long period. And finally, we have the transformation where the virus integrates into the host's DNA, often impairing the function of tumor suppressor genes. And this transforms the normal animal cell into a tumor cell, leading to uncontrolled cell division, ultimately cancer. Now, this is the viral growth versus a bacterial growth curve. So we need to understand how the curves look generally. So over here you see that the viral growth curve, which is a one-step growth curve, so there is an eclipse period that is the lag between the initiation of the infection and when the first intracellular infectious virus particles is produced. So during this time, what happens, the virus has uncoated and its genetic material is being transcribed and translated to create new viral components. So no infectious particles can be detected if the cell is lysed at this stage. Then there comes the latent period. This is the time from infection until the first virus particles is released from the host cell. The rise period, which is the burst. So a rapid increase in the number of infectious virions as the host cells burst and releases the newly assembled viruses, and the burst size is the total number of new viruses that is released from a single infected cell. If you look into the bacterial growth curve, which is a lot more like a binary fission, so the right graph is basically of the bacterial growth curve, which reproduces through binary fission, where one cell divides into two. You have the lag phase where the bacteria is adjusting to the environment. They are metabolically active but yet not dividing. Then you go into the log phase, which is the more exponential phase, so it's dividing at a constant maximum rate. This is a steep upward curve, part of the curve. The stationary phase, where the living cells, where the growth stage slows down, the rate is slow and the nutrients are depleted and the waste products start accumulating. And then you go into the death phase, where the number of living cells decreases exponentially as the conditions becomes toxic. Now there are two types of viruses which can be there. One is the equilibrium viruses that have been associated with the host system for a very long period of time. They are usually non-lethal but spreads well, like the common cold viruses. And the non-equilibrium viruses have recently jumped from another species. They are often lethal, may spread poorly or well, represents most of the deadly viruses. Okay, so these are some of the examples of the different viruses, like you have polio, common cold, measles, and herpes for equilibrium viruses, and for non-equilibrium viruses you have SARS, influenza, HIV, Ebola. So number of human coronaviruses which causes common cold with mild flu-like symptoms, they are examples of the equilibrium viruses. Now coronavirus SARS-CoV-2 is a non-equilibrium virus which has jumped from another species, most probably bats. Now, if we start linking the viral infection with cancers, you can see that viruses known to cause cancer are called oncoviruses. Now, oncoviruses trigger cancerous transformation of cells by encoding proteins that impair the function of tumor suppressor proteins. Tumor suppressor protein P53 is referred to as the guardian of the genome and controls cell cycle progression and also initiates programmed cell death in response to DNA damage. However, when P53 is inactivated by the binding of an oncoviral protein, it can't initiate programmed cell death, and therefore DNA damage persists that leads to the development of cancers. Now, from the point of view of virus, hyperproliferation and the lack of programmed cell death is important. Now, understanding the microbial pathogenicity and infection is important. So, relationships between two organisms can be very complex. A larger organism that supports the survival and growth of a smaller organism is called the host. So, any organism that causes disease is known as a pathogen, and its ability to cause a disease is pathogenicity. So many bacterial virulence factors that facilitates the survival are encoded in unique sequences of DNA that are readily swapped between the bacteria through the horizontal gene transfer. Okay. So there can be multiple virulence factors that you see over here. So what can be those? So there can be like internalin A, so it's a primary virulence factor. It binds to E-cadherin, a receptor on the host cell membrane. Then E-cadherin interaction, where it's binding the internalin A to the E-cadherin, which triggers the signaling cascade involving host proteins like the alpha-catenin, beta-catenin, actin filaments, and then endocytosis. So like it's accumulating in the zippering of the host cell membrane around the bacteria, leading to internalization into the vacuole, the phagosome. Now we will talk a little bit about the channel forming or pore forming toxin inserts into the host cell membrane, forming a channel pore. So pore forming toxins are virulence factors secreted from pathogenic bacteria through insertion, then channel formation or pore formation that spans the lipid bilayer. Then there is an osmolarity shift. So the pores creates an uncontrolled path between the intracellular and extracellular environment. So water enters the cell due to high internal osmotic pressure, and there is cytoplasmic contents. So cell lysis, the influx of water causes the cells to swell and eventually burst, leading to cell death. Now T cell activation of superantigen stimulates massive amounts of cytokines production that result in shock and death. So the best studied superantigen is the staphylococcal enterotoxin B, which exhibits superantigen activity at a nanogram concentration. So again, getting into the same things like this, we will now talk about the yes. So the thing that we were discussing about is the exposure and transmission of infectious diseases. So there can be airborne transmission, that is through dust or droplet nuclei. So like measles, tuberculosis, chickenpox. Then there can be contact transmission, which is the most common way infection spread and is divided into like two categories. That's the direct contact, like a physical touch between an infected person and a susceptible person, There can be an indirect contact that's touching a contaminated object or surface known as a fomite. There can be vehicle transmission like cholera, saphenmolysis, hepatitis A, blood or plasma. There can be vector-borne transmission like the biological vectors, mosquitoes, um, carrying malaria or Zika virus. There are mechanical vectors like a housefly landing on food after being on waste. Then vertical transmission, that's passed from the mother to the offspring, so it can be through and across the placenta during pregnancy, during childbirth, etc. Now, some microorganisms can establish an infection with a very few in number. Conversely, others microorganisms are required in high numbers to establish an infection. So, infectious dose, that's ID50, is the number of microorganisms required to cause clinical disease in 50% of the inoculated hosts. And the lethal dose 50, that's LD50, this value refers to the dose of or number of pathogens that kills 50% of an infected experimental group of hosts. Now, there are general characteristics of antimicrobial drugs. So, some of them is the selective toxicity. So, selective toxicity is the ability of a drug to kill or inhibit the microbial pathogen while damaging the host as little as possible. So, drugs that target the specific structures or metabolic pathways are unique to the microbe and are absent in the human cells. Now, therapeutic dose versus a toxic dose is that the drug level required for a clinical treatment of a particular infection. The toxic dose is the drug level at which the agent becomes too toxic for the host. So, this is the ratio of the toxic dose to the therapeutic dose, known as therapeutic index. So, what do we mean by narrow spectrum versus a broad spectrum? So, narrow spectrum is effective against only limited number of pathogens, variety of pathogens. It's preferred when the killing of an infection is known to avoid killing good bacteria. Whereas for a broad spectrum, it's effective against many types of pathogens. So it's used for a specific cause of an infection that's unknown. So what do you mean by acidal versus static? So bactericidal, the drugs that kills the target pathogen. Bacteriostatic, the drugs that inhibit the growth of that pathogen. So once the drug is removed, the bacteria may start growing again. So we need to rely on the host's immune system to clean this mess up. And this is the bactericidal action of penicillin. So the penicillium mold colony secretes penicillin that kills the Staphylococcus aureus strychnia. So the cell wall synthesis inhibitors, so you have the penicillin G, that's high activity against the most gram-positive bacteria, low against gram-negative, destroyed by acid and the penicillinase. You have ampicillin, that's active against gram-positive and gram-negative bacteria, it's acid stable. And the methicillin, that's penicillin resistant but less active than penicillin G, and it's acid labile. These are some of the cell wall synthesis inhibitors. Protein synthesis inhibitors, you have the tetracycline. So, three members of tetracycline family. So, tetracycline lacks both the groups that are shaded. So, chlorotetracycline differs from tetracycline having a chlorine atom. Doxycycline consists of tetracycline with an extra hydroxyl group. Now, there can be inhibitors of nucleic acid synthesis, that's norfloxacin and ciprofloxacin. So, it inhibits DNA guidase and topoisomerase 2, thereby blocking the DNA replication. You have rifamycin and rifacillin, that's inhibiting the bacterial DNA-dependent DNA polymerase. And there are antimetabolites, the trimethoprim, dapsone, isoniazid. Mostly they are bacteriostatic. So isoniazid, it inhibits the mycolic acid synthesis. There can be antiviral drugs. These are probably the most publicized antiviral agents like the Tamiflu. The Tamiflu inhibits the viral molecule neuraminase, which is essential for the release of newly synthesized influenza A virus particles from the host cells. Thus, Tamiflu received much attention during the 2009 to 2010 H1N1 influenza pandemic. Now, the mode of anti-HIV agents where the infection begins with HIV fusion. So, the fusion inhibitors blocks these steps. So, once inside a host cell, HIV uncoats and its reverse transcriptase forces the host to make DNA from the viral RNA. Now, the viral DNA is then transcribed and translated into a linear polypeptide that is cut to release the viral proteins. And the viral DNA is added to the host DNA by the action of a viral integrase. Now, for the first step, the fusion inhibitors blocks this particular step. The RT inhibitors blocks this step, so there can be reverse transcriptase inhibitors. You have some protease inhibitors that inhibit the production in these viral proteins, and the integrase inhibitors blocks the last step. Finally, coming into the antibiotic resistance. So what we understand over here is that, so in a normal scenario, the antibiotic enters the bacterium and binds to a critical enzyme. This binding blocks the enzyme's function, effectively killing the bacterium or stopping its growth. Now, what happens? There is an altered critical enzyme. So the bacterium undergoes a genetic mutation that changes the shape of the target enzyme. And because the shape is altered, the antibiotic can no longer lock into the enzyme, and the enzyme continues to function and the bacterium survives. Now, the protein that degrades the drug, so the bacterium produces a specialized enzyme designed to break down the antibiotic. So as soon as the antibiotic enters, the protein chemically degrades it into harmless pieces. The critical enzyme remains untouched and functional. And finally, the bacterium develops a pump into its outer membrane, the efflux pump. So while the antibiotic can still enter the cell, the efflux pump immediately identifies it and stays busy pumping it out. So this keeps the internal concentration of the drug too low to cause any sort of damage, and that is how it's resisting to the antibiotic provided. Okay, so yeah, that's all from my side about the lecture. Okay, so we will have, if you have any questions, you can ask me. Maybe 2 to 3 minutes. If you have any questions on any part, you can ask. Then we will start with the quiz time. No questions? Okay, then I think we should go into the quiz thing. Okay, you people can answer me. So this is a match the following of mode of action versus the antibiotics. So what does these individual antibiotics do? How they act? So it's the mode of action of the antibiotics. So yeah, you can answer me. So the correct answer is, so what does norfloxacin do? It's a inhibitor of DNA gyrase that's essential for bacterial DNA replication. Ampicillin, it's a beta-lactam antibiotic that interferes with the synthesis of the peptidoglycan layer in bacterial cell walls. You have the rifamycin that's blocks the transcription of DNA into the mRNA, and the tetracycline that binds the 30th subunit of the bacterial ribosome, preventing the attachment of tRNA and thus halts the protein synthesis. So A2, B3, C is 4 and D is 1. That is option 2, 3, 4 and 1. That's option B. Okay, so the next one. Which of the following statements are correct? Which of the following statements are correct? So statement 1 and 2 is incorrect because both of them says that they are the only ones individually, which is not true as both of them have a prokaryotic origin. Statement 3 is correct while 4 is incorrect because modern biological evidence suggests strongly supporting prokaryotic origin. Okay, so only option just because option 1 and 2 says that individual things, so we cannot consider them, so only option 3 is correct. That is option C over here, right? Now the next two, which of the following statements is or are true for gram staining? So crystal violet is the primary stain used from the gram staining method. That is true. It is based on the properties of the cell wall. That is also true. It is based on the properties of the bacterial cell membrane. Now, this is primarily not very true. And the last one says safranin is the primary stain used in gram staining method. I'm also not okay with this because it's a counter stain. So crystal violet is the primary stain. But looking at the options, C is more appropriate, where you have 1, 2, and 3. Okay, so it will be option C. Now, in a virus growth curve, eclipse period denotes the phase when. So, what does this eclipse period denote? So, what is the eclipse period? It is the time between entry of the viral nucleic acid into the host cell and the appearance of first completed infectious progenivirus particle. So in this period, virus proteins are synthesized. That's true. Virus are replicated. Progenivirus particles are assembled. Yes. So all of these options are true. So it will be option D. That is all of the above. Yeah, that is correct. Now, bacterial cell wall is composed of. Very easy. So what is the bacterial cell wall composed of? Very direct question. So the bacterial cell wall made up of a peptidoglycan. Peptidoglycan is a combination of sugars. That's the NAG and the NAM and amino acids. So like the D amino acids, D-alanine, D-glutamic acid, right? So it's a combination of sugars and amino acids. That is option A. Which of the following statements are true for equilibrium viruses? So we spoke about equilibrium viruses. Which of them is true? So equilibrium viruses are associated with particular host species for a long time. Usually they are not lethal, but they spread easily because they have adapted to the host. They typically cause mild or manageable symptoms, and they are long-term parasites of the host species. So they have a stable long-term evolutionary relationship with the specific host. And the first two options are with the non-equilibrium viruses. So it will be 3 and 4. Right now, okay. So during the second wave of COVID-19 pandemic, some patients died due to cytokine storm. Which of the following statements are true for cytokine storm? Maybe try answering it, but what you mean by a cytokine storm, that is important to understand. So, a cytokine storm is a severe immune reaction in which the body releases too many cytokines into the blood too quickly. So, in the context of severe COVID-19, so that happened due to the hyperactivation of the T lymphocytes by a super antigen leading to release of massive amounts of pro-inflammatory cytokines. So that is option A. Okay, question 7, that's question 8. So, bacterial virulence factors are unique DNA sequences and are swapped by. So, how are these unique DNA sequences swapped from one bacterium to other? So, we talked about the horizontal gene transfer, the three mechanisms: transformation, transduction, and conjugation. Now, the next question is which of the following statements is incorrect for HIV? You have to find the incorrect statement for HIV. So it has a double-stranded DNA as a genetic material? No, it's a retrovirus, meaning its genetic material is a single-stranded RNA, not double-stranded DNA. Shows reverse transcription, true, infects CD4 plus cells as their host and causes AIDS. Those are all fine. So option A is incorrect. And last one says oncoviruses cause cancer by. So how do oncoviruses cause cancer? Yes, by encoding the proteins that impair the function of the tumor suppressor P53. That is correct. Okay, so right. That's all from my part. That was the last question. So if you have any questions, you can go ahead, ask me. No questions? Okay then. Thank you very much for joining. We will again meet in the next week on Friday with a new set of lecture. Thank you.